Does everybody keep a song, like, is there always a song stuck in your head for anyone else?
Oh, I get songs stuck in my head all the time.
Like, just, there's always something singing.
There's always something.
There's always something. It doesn't necessarily have to be music.
Okay.
It could be like a phrase.
Oh, interesting. Okay.
Yeah, sometimes it's like a phrase or a word, like a really good word.
Huh. A word with
So, like,
good mouthfeel, probably.
Yeah.
So one of
or
your
just a
brains
weird
is
word.
just like got a mantra and it's got meditation going on.
Yeah. I feel like it's closer to being like tortured by demons than meditating, but you know. Sure.
Just change your perspective.
That distinction is a relevant one.
Glass half full, I guess.
Yeah.
I had about 3 months one time where Prince Ali from Aladdin was stuck in my head.
Oh, yeah.
Every single day.
3 months?
That's a tough one.
Oh, it
That's a
drove me
sticky
nuts.
Yeah.
Well, there's a fish up ahead by that rock.
Ooh, yeah.
Apparently that was a memorable 3 months. How'd you finally dislodge it? Like, what was the thing that replaced it?
I don't remember.
Huh.
Just one day it was gone.
One day it was gone.
And you're like, oh, thank goodness.
Go presume?
Yep.
Go presume?
I hope this conversation doesn't restart it.
I hope not to.
Does no one sing it?
It might start it for me.
Oh,
Oh, look
that's
at how
a pretty
iridescent it is.
Yeah, it's a little green eye, chlorophthalmus.
That's beautiful.
Get their big green eyes. So cute.
I've definitely got Prince Ali going in my head now.
It did start up as soon as he said it.
Yeah. I mean, it hasn't displaced what was already in my head, but is now playing right alongside Bright Side by the Lumineers. Simultaneously.
Oh no.
Go wide.
Going wide. That's a remix. I don't
It
think
is
the
a
world wants.
weird one. It's like some like jolly, slightly like Middle Eastern sounding music and like a very grimy guitar riff.
So
It's a good
just
mix.
a heads up, Gabby, we are about 1.5 meters from our next Niskin.
Okay. Thanks for keeping me on track. I was very dangerously close to going off the rails there.
You'll be right at home here.
Someone just commented about what you said, Chris. I have that too, and sometimes it's half of a line of a song, and you don't even know the rest of the.
Oh, that's the worst.
Oh, that's
Oh, I
yeah,
never have a whole
totally
song.
is,
It's only
yeah.
a section of it.
It's the where's that from?
Uh-huh.
Okay, you ready for this?
Yep.
Okay, Mary, we're getting ready to take that Niskin.
Yep.
And we're actually on the downside with your CO2, so we're going to be immediately above it. We're going to have a nice
Are we retracted?
Yeah,
bracketing
we are
of Niskins above, in, and below the CO2 peak.
That sounds good. The CO2 has some response time, so we'll probably... It sometimes is really deceptive. You think it's on a downward trend, and then you're like, oh, we're going up again. So, but yeah, we may be exactly what you're saying,
this
on
3
either side.
that we're doing now?
Should be
Yes,
Niskin
it'll
3,
be
I think. Yep.
And data just as well, I've now finished pump 1 as well.
Okay. One moment on that.
Yeah.
Got
That was it.
You got it. It fired.
I've stopped grabbing them because that's how I got that like Niskin to trigger that we did not mean to trigger. And it's
Yeah.
working better now.
Yeah, I saw one of the other watches. They pulled the little bubble off, but it
Yeah.
took a while to work out what it was. I was sitting with Sean watching it, and it was like, really? The pilots have a stress ball for their manipulator now? It's like,
Yes,
Yes.
that
it's
is
just
what
like a
that
thing
is.
to...
That makes a lot of sense
It's
to
just
me.
like, yeah, it was just sat there, like
feel like the
beautifully
craft
held
needs
within the
it.
claws. It was just like, oh yeah, I can just squeeze a little squishy blue ball. It's like,
The craft heard that it's getting replaced.
yeah.
It's freaking out.
And it's just got a little song in its elbow, and it's just like trying to work out what is this. That's how
I
it
feel
stays
like the crown
calm.
is ready for retirement.
Yeah.
Like it really wants to be holding a pina colada.
It wants to be with me, being a beach
Oh
otter.
my gosh, that's what
Wants
its
to be
retirement
facing-
plan is. Is it gonna serve drinks in the billionaire suite?
Oh
Oh,
no
yeah.
no! I got the great plan. It can come and like I can attach it to my Adirondack chair,
Oh, yeah.
so then it can feed me my breakfast off my belly while I'm
Quite
sitting
frankly,
facing
I
the
don't
sun.
think operating that arm is a great retirement plan. Like,
I wouldn't trust it to be
I'm not going to be
anywhere near your
operating it. I'm going to
face.
be retired. I'm going to be telepresence,
I'm just
baby.
thinking about the carpal tunnel.
I'm going to have somebody remotely operating it. And just
We're
Maybe-
gonna be
serving
on
me-
the ship
Yep.
serving me
operating
That
the
was
arm
a
at Chris
252.
German's like lakefront.
252.
Yes.
You said
Did
it's
I
ROV
miss
training.
Gonna be like
251?
freshly sliced
251
papaya
was-
slivers.
Was it with
Is it with
the
the multiple
2?
3?
Oh, was it at the same time as 250?
No, it was shortly after.
Okay.
It was at 11:51.
So that was when it started.
Yeah,
Yeah,
for 25. For pump number 3.
Okay.
And pump number 1.
Yes.
What was the end time?
End time was 11:56.
Oh,
Right.
If the
Would you
raptor
like
loses-
to sit and smell
And
it?
Sure. yeah.
depth was 720.5.
I'm not quite sure. It might be a little bit steeper than you did last time.
Okay.
You just kind of have to
what was
stay
the start depth?
ahead of things
Start
until you
depth
get the feel
859.
for the rhythm of it,
Yeah. Okay.
and then you can start
And
to
what was
cut
the
it a little
volume?
closer.
120110.
Okay, thank you.
And Chris, it looks like you were correct. It's not increasing again. It's pretty much finished its downward trend and hasn't gone anywhere. So hopefully some interesting chemistry between those
Mhm.
samples.
And are you keeping any of your autos?
Yeah, so whatever you like, you're welcome to use it or not. I think you've been using it, so I would stick with it while you're getting a feel for things, but stay sort of aggressively out front until you get a feel for how fast you need to go.
I feel like we haven't seen anything on this dive with the kind of density and diversity of biology that we were seeing much deeper.
And there's some
Yeah,
more
I
that's gonna
agree. I
send you
was,
forward.
Bridge.
but we
huh?
have also been in a more. I think it's
I guess
been
we've
pretty
seen a lot more
steep.
talus and we haven't
Yeah.
seen much of the cliffs.
167.
Yeah,
Because the other places we'd come up a cliff and then there was a plateau that was...
yeah, we haven't had the plateau and the boulders on this dive yet.
I have a really great question here.
Mm-hmm.
What do you guys do when it is your schedule shift, but the ROV's not in the water?
Uh-huh.
We launch a different robot. We have a backup robot with Sentry.
Mhm.
We do.
So I'm the science communication fellow. Usually we have scheduled schools or groups that would like to do ship-to-shore interactions. So that's some of my tasks, and yeah.
Yeah, in my case, it really has been that for the last week at least while we've been at this seamount, if Herc wasn't diving, it was normally because Sentry was diving instead. So Mary's helped us rig a thing where we can actually get live data streams just through acoustics from Sentry, so I can actually see what it's discovering as it's doing those surveys. So I've been sitting down in the little the Sentry cocoon, the little Tetris pack that is how do you get yourself into an extra seat where there's no seats left. It's cozy down there.
And for me, when we've got two vehicles, there's a lot of, we had a problem in the first week where one of my sensors stopped working. So now every time either Herc or Sentry ends a dive, I have to take the one sensor that's left and swap it over and set it up again. And so then yeah, and then when the sentry's in the water, there's some amount of, watching the data come through and setting up that stream. And then also data analysis, trying to see if we've seen anything interesting in the data we've got from the dive so far. And then sometimes sleeping, occasionally.
Mary's become our pit stop specialist.
Yeah.
I think she's going to get a second life doing tyre changes.
Esteban and I were saying exactly
Tyre
the same.
rotations, you
We've
can
got
just move
really
that
good
fast.
at it now.
Yeah.
It's like, it's gone from being like an hour of like trying to work out how we're going to do this to being like 15 minutes rapid changeover.
It's a lot of anthomastus.
Look at all those, yeah, look at all those mushroom corals.
And our next Niskin is going to be at 680.
Yeah, when we're not diving, as a navigator, I set a mapping watch, or I am standing by for sentry navigation assistance as well. So I always have my watch. I just sit in a different chair, slightly warmer chair
Another different
downstairs, and either map with the multi-beam and do some data cleaning and processing, or make sure the sentry team has what they need and monitor our USBL tracking. Are you talking to me? Oh, I can't hear you at all.
Funny. Can you hear me better now?
Oh, yeah.
Okay. In lower, there's a cool view of just like how many. What are these?
All the mushroom corals,
Yeah.
anthomastus. I know Santiago said he wanted some of these.
Yes, please.
Okay.
He's here?
He's
Oh
back.
my god.
Disembodied voice.
Is he really?
He is,
Yeah.
yeah, he's back in the studio.
He's an hour late for his 4-hour early watch change.
Oh my
Yeah,
god.
I'm sorry.
Santiago, you'll be really pleased to hear that our Niskins were in the middle of 2 different CO2 features.
So would you
and
like
then
to get
the
some of these corals here, Santiago?
Yeah.
Yeah.
Yep.
Is
And
that what you
then
guys want
Are
to do?
we between ship moves?
Yeah,
at
the
moment?
ship is stopped and Atalanta is settling out from her moves, so
Perfect. Let me
we are stable
find
here.
a spot.
Have you seen anything else?
There's been a ton of these.
Yeah.
But
It just all
But
of
I
a
think-
sudden got super dense with
But this concentration
anthomastus.
is huge, isn't it?
but anything else?
Not really.
Really?
There's like brittle stars, lots of shrimps.
Hmm.
It hasn't been anything like the densities we were seeing
Yeah, not like
on the
earlier
previous dive.
when we were deep.
Has this been mostly this talus terrain or a mix?
It's been going between this
Intermittent.
but even when there was like proper outcrop, still
Huh.
it wasn't like we were seeing further deeper.
Interesting.
We weren't seeing all the
Just
corals
trying
and
to-
things.
And you landed at
Stopped at the base of a
900
really
or a
cool
bit
feature.
shallower?
Yeah, okay. Do we want to be up there
we landed
more, or-
at
That looks, I mean,
something
I
like
can sample here.
850.
Yeah.
It's fine.
850?
It's just after this, you
859,
should
yeah.
go up
Yeah, okay.
the stairway there.
Yeah, I think
Okay, we
Adam
are
had.
stick
I think
locked
Adam
right now.
got,
Stairway to
went back
heaven.
in at waypoint 5
I
and
don't know how
Yeah.
stable
was at
we are,
waypoint
but
6 when we took over. So
Yeah, we're almost at waypoint 7. Well, her just
how
passed
many of
waypoint
these
7.
did you want?
Just
Just
serve
enough to
few
make a
of 'em?
bouquet?
Yeah.
So we'll probably scrape and slurp these.
Yeah, I think
They're
it's
the
a good
flowers of the deep.
idea. Have they all been like these small, or have you seen big ones?
There was some bigger ones earlier.
Yeah, well, when we were deeper, they were bigger.
Yeah,
Wanna
yeah,
go
yeah.
for zoom video?
But that might just be because they're older.
I'll just get a zoom of
There's
these.
some larger ones, and there's some really, really small ones.
There you go. Okay, go wide.
Going wide. And Asako asks what these are.
The anthomastus.
Yeah, the pink things.
Okay, I'm just getting here. Rocket cam. Science, do we know which jar is open?
I think
Yep,
they're
one
all
looks good.
Oh, that's
Okay.
right. We
Yeah,
Are
we
we back
started
to-
over.
did a
Redo.
We're back to
refresh.
all of them.
Back to all
Yeah.
of it, yeah.
In some ways, it was a convenient break because we also got the eDNA sampler on.
Yep.
We got replicates of water column data, which meant we were able to pick out really nice places for Niskins. And we got to offload that boulder
And get a bigger one.
and get a bigger one.
It definitely was a boulder. Do you guys weigh them in the lab?
No, we don't.
It's always fun to try to weigh things on a ship.
Yeah.
I'm like, oh wow, I'm 20 pounds lighter, 20 pounds heavier.
Yeah.
There we go. And go for Zoom video.
Going for Zoom. Alright, suction.
Yeah, go for suction. And how many are we looking to get, Santiago?
Just a couple, maybe 3 or 4.
You're very quiet, Santiago.
Oh.
Unless my ears are blocked.
Sorry. Is that better?
Yeah.
Yeah.
So,
I'd say probably a couple, maybe like 3 or 4.
okay. I saw one go in so far.
Yeah, same.
Oh,
That's
two.
two.
Okay.
Maybe the corner is closer to you might be a
Easier.
Oh, cool. Good idea.
easier
If
for the
Adam were awake,
scraping.
he'd say, you know, you could just pick up the rock and bring them all.
No,
By
you don't know
catch.
where this one came from.
You don't know where it's been.
Well, it can't have come very far because we're nearly at the top of the hill, so... It's a very popular rock. All the corals
All
like
the
it.
rocks seem to
Good
be
real
popular
estate.
around here.
Well, no, not really. The corals like these ones better.
Oh, maybe
Your
that one
CO2
wasn't
sensor isn't
easier.
doing anything interesting by any chance, is it?
Right now, the CO2 sensor is basically baseline. And the same baseline we're observing at seamounts
I
pretty
was
far
just-
from the vent.
Wondering on that point of this talus looks just like all the other talus, then it's a, what's the underlying principle of why does this suddenly have the
I'm,
high
like,
concentration
really
of corals?
impressed.
Yeah, I know,
These
it's
guys.
not doing anything
I'm not
at
out
all.
of stick lock
No.
yet. I'm just gonna
I feel
see
like
how-
it would also
Do you want me to
be-
get out of stick lock?
Maybe we'd want to see a local dip rather than rise. But I'm not seeing that either. And there's no anything interesting in
No.
temperature or salinity.
No. I was just looking at the same thing. It's
There we
Okay,
go.
I
there
think
we
that's
go.
about 4-ish.
Yeah.
What do you think, Santiago? Do you want more?
I think that looks good. Thank you.
Okay, sounds good. You can stop suction.
Copy. Suction is off.
Do you see them in the jar?
We
Oh,
will
so
when we
we're
stop
just-
suction.
Yeah, suction's off.
Okay.
We're just waiting to see him.
Did you? I thought I saw them go in.
I definitely saw stuff go in. It's
Did
definitely
you try
open.
reseating the jar?
I can try that.
Yeah, go for it. You can turn suction back on.
There we go.
There they are. Yeah. All right. It might just be a little floaty.
Where did it go?
I think they're just
They're-
at the top of
Yeah, I think they're just really floaty.
yeah. but we saw at least 2 of them.
Mm-hmm.
We can always take a look in a bit and see if they've sunk down. But
what do you guys think? Do you want to take a few more in a different sample jar, or would you like to call it good?
What do you think, Santiago? Are you happy?
Yes, I can hear. I don't know. Maybe, yeah, maybe we can give it a few minutes, just do something else, and then check. I'm sure we'll see them again. Well, actually, I'm not sure, but
Okay.
Well, do we
likely.
need to put it in a
I would
different
be
sample
pretty
jar?
confident that
Okay.
this is the dominant fauna here.
Yeah. We can keep them in the same sample jar if there's...
Yeah, we don't need to put
Okay,
it in a
you don't
different
count as
sample
one
jar.
sample.
We haven't moved the ship.
Okay, so do you want...
Let's get 2
I
more.
wasn't clear.
Let's
Okay.
get 2 more since we're here and we're set up, just to be
Yeah.
absolutely sure we've got what we
Okay.
need.
copy. Alright, suction come on.
Thanks, video.
'cause normally with a
If you
floaty,
read my
they'll-
mind any better, I would be creeped out, though. Just notes there.
Just a warning.
Yeah.
there's
Yeah,
one.
that definitely went in.
I wonder if it's getting stuck to the mesh up on the top or...
Can you pan down? There's
Yeah.
an angle that works well, and it's, yeah, it's more downward. More a little? I'm going to go for
That
this
one?
lower one. Yeah.
Yeah.
Mm-hmm.
Down is the best angle here.
Get him, Gabby.
Okay. That one gets to live another day.
That one's
Mhm.
strong.
Oh,
Oh.
some
Getting
bonus
some other stuff here,
material.
too.
I
Okay,
got it.
that's 2 more.
Nice. Okay.
There it is.
We
I saw
saw.
that one arrive. Yeah.
Yeah, we definitely saw
Great.
that.
Thank you.
And there's a shrimp having a little disco party in that hole with a laser.
And Megan, that is 253.
253.
Okay. We wanted to look at this feature, right?
Yeah, I personally wanted to look at that feature. I don't know if everybody else does. Just seemed like it would be a cool thing to follow up.
And hopefully that...
I think we can probably get a ship move going, unless
Alright.
there's some more sampling or something to do around
Nope,
here.
I think we're... What was our next Niskin due? 680?
Yep, 680.
Bridge now.
So it looks like there should be a nice ridge to follow all the way up to waypoint 8 on the high-pack
We'd
survey.
like to move 20 meters, 157.
Oh, I see. There are rocks.
So many anthomastus.
This is so pretty.
This is...
It really is. The colors are amazing.
Mm.
Oh, thank you. That's,
We are also now getting some decrease in CO2. So
It's continuing to fall.
it's just started to drop in a
Mhm.
sort of more local way.
Let's look at that big shrimp.
Sure.
Is it unusual, Megan, to see this
Go
higher
for zoom
density of
video?
just one species of coral without any others?
I don't know if it's unusual. Maybe it's a little unusual.
We're almost into your depth zone, right?
Yeah, it's a heterocarpus.
I would say it is unusual, yeah.
Yeah, it's a bit unusual. I mean, I have come across different areas with super high densities of anthomastus or pseudoanthomastus on other seamounts,
Mm-hmm.
but usually there's other corals too.
Right.
But, you know, tons of these guys tend to show up in different areas.
I guess, did any of you see the
Alright, go wide
area south of the vents where they were really abundant? This is,
Yeah.
I would say, this is
Oh,
light
yeah.
abundance.
That was,
I
yeah.
feel like this is the most abundant coral that we've got
Yeah,
here.
you got to you got to do the exploration that went south to the other waypoints, didn't you? I remember watching it on the video after I left the van.
Yeah, that was crazy.
Yes. Yeah, this is very similar to that, isn't it?
Santiago, what is your thinking for the final two Niskins, or final two? Do you want them in abundant regions or particularly interesting species or water column?
Yeah, that's strong for NA.
Well, are we going to do the base jump down and come back up the hill? Because we could do the same depths as before.
Yeah, I hope so. But
I hope so too. Yeah, I would save them for
Save
a
them?
little later.
okay, that sounds good to me.
We still have like 200 meters to go to the top.
Yeah.
Vertical.
But we can see the final waypoint on our map.
It's 265 meters to waypoint 8.
Have you seen any anemones?
Not
One big
recently.
fat one.
Oh, there's a squat lobster right between your lasers.
Yeah.
There was one I think it was it wider than the 10 centimeters with the lasers? That big fat anemone we saw, Megan?
Oh yeah, no, that big
Yeah.
actinostolid was like definitely
Tilt's
way
running
wider.
away, doing crazy stuff.
Oh yeah, you were saying
Sorry?
dinner plate size.
Yeah, it was like
The
a dinner
tilt
plate
is
size.
running away and doing crazy
Oh,
stuff.
okay. Yeah, okay.
But stay out front
Yeah.
just to
I'm
manage the tether.
Getting up there.
This is really pretty. Okay, I admit, this is a little more unusual than I was anticipating.
Just
It
this.
just keeps going.
And
I feel like they get denser as we keep going up.
Mm-hmm.
don't stop for zooms until you just start a little bit
Yeah,
more
yeah.
out front.
There's a big rock up there.
But given that inside the crater we get a lot of anthomastus, this might be where
Look at
they're
this
seeding
guy.
from.
Look at that rock there.
Oh wow, yep. It's like, this is the local high, and just completely coated.
There we are.
Nice.
There should be some brittle stars coming up. Oh yeah.
There's a sandy bit with the black.
Mhm.
I think it's deeper here.
You want to try a
we could try.
push
See if we
cord?
can get a push call.
Yeah.
Yeah.
Cool.
There seems to be a lot
Yeah.
of sand.
Get out
We're
front.
not moving. Yeah. Okay.
We're
I'm
not
gonna
moving. Analyst is almost done with her move.
Copy. I'm just gonna get up a little bit.
vector and low is amazing.
Yeah.
Moving over those riffles at that height, it's just like...
Yeah.
Okay, we want to try push core here? Let's do it.
Yeah, go for it. Set her down. I'm probably just going to stick the jaws in over on the left-hand side, just to see if it's even worth getting a push core out.
Mm-hmm.
Alright. Locked.
No. Doesn't seem that deep, does it? Oh, maybe it is deeper.
See, this is what the temperature probe should be doing.
Yeah, the pokey
Oh, I'd
stick.
get in so much trouble, guys.
That looks...
I don't know, that's like a few inches, 2.5 inches.
Do you think you got all the way down, or was it just pushed back from the
I
sediment?
don't know, I guess I can...
I would... Let's give it a
I'm
try.
not going to say no.
Let's give it a try.
Okay, sounds good.
It feels like that's thicker than it was where we tried before.
Yeah.
It is deeper than where we tried before. I don't know by how much.
And I've also just realized that we were planning to take our Niskin at 680 and we're at 677. So this seems pretty convenient.
Perfect. I'm just remembering that you told me 680.
We can definitely do both.
I'm going to bring this one up front first, just because it helps to know
Yeah.
what I'm dealing with. Go for it.
Well, it's going.
Mm. That's quite satisfying.
Yep.
What do you think? Shall we give this one a try?
Yeah.
Yeah.
Yeah, definitely.
Okay.
all just came up.
Yeah, I think it's kind of coarse.
Yeah.
It came out that fast, hey?
Yeah.
Yeah,
Okay, that's not even going to make it through
No.
no.
a starboard sample.
Okay, let's shake it loose. Stow the corer,
Yeah,
and
it's
we'll try
totally
again
empty.
when we get up to waypoint 8. If that's going to be on the flat surface, it might be even thicker. And maybe...
More sticky?
Yeah. Because I think the black fragments are the ones that have been lofted from the explosive volcanism, but it's whether there's other stuff around to actually help bind that together. Because I think Adam can pick out the black bits from the other stuff, but if it's just the black ash, then I think it's always going to be just like gravel.
Yeah.
Excuse me, I would like you to halt. Halt. All right.
So Mary, would you like us to go downhill 2.5 meters for the Niskin, or is this close enough to 680?
Let me just come have a look at the CO2 data and see if there's anything there to suggest one way or the other.
Okay.
So it looks like we might be on a slight upward trend right now. So I suggest maybe we could go up to 677, get clear of the sediment and fire there. Well, we could also just fire here. It's a location we know we had an ORP hit. So, and the sediment settled fast. Yeah, let's fire here.
Okay, sounds good.
Thank you.
Ryan, can
Oh,
you
we're
set,
gonna do
set me
Niskins?
up for a Niskin?
Yeah, absolutely.
I'll grab the cameras.
Yep, that's Niskin 4.
Yeah, you go.
Niskin 4, thank you, Data. And we're retracted?
We are fully retracted.
Is that it?
I didn't see it.
I didn't
Did
see
anyone
anything
see it?
move. Nope, I didn't see anything move.
I didn't either.
I can't tell because it's all whited out. Can we secure the light?
Yeah, let's do that.
Okay, here, what is that? Starboard port. What? 1, 2, 3. I don't
It
think
looks
it's gone.
open.
Okay.
Mm-hmm.
But
It's
it-
open to me.
I think if you go anymore, you're gonna...
Well, I'd rather get the sample. I'll do it by holding the ball, so at least we won't lose the ball if we break it. Yeah, it does seem like it's not triggered.
It's not.
I think I'm going to risk the wrath of... Actually, it's probably going to be us that has to fix it, so... Okay, you guys watching?
Yeah.
Yeah.
Seems like something's wrong there.
Yeah, nothing's.
There's no way that's right.
No, that's way too. I mean, we just routed these the other day, so
Yeah.
it must be caught in the mechanism or something.
Rats.
Do you want me to try? Like, if you stow the arm, I could try moving around and re-landing, and maybe the bump from the vehicle might.
Could just get up and sit down again.
Yeah.
Go for it. I like that idea. I like it better than trying nothing.
Okay. Do you want to keep the arm there, or?
I mean, if you're okay with
I'm fine with it. Okay.
I'll keep an eye on
Just
the
go
end cap.
straight up and then straight down again.
Yeah, I'm afraid if we trip a different one, four
then we'll end
will
up
go
2 will,
soon.
yeah. It must be kind of...
Nothing there.
Didn't see it.
No. All right.
Okay. I feel like
That's
I'm
fair.
okay with us breaking this off and having to reroute it if we can get the sample. So
Okay.
I'm just going to go for it.
Still
Rats.
nothing.
That stinks.
Yeah.
Where do you want to put the ball?
Okay, the ball will go in the forward,
Bow box.
one of the forward bio boxes. I don't, unless data says different, I don't think it's going to matter which one.
No, either one's fine.
Okay. Forward box coming
Yeah.
open.
Lotto balls are not famous for eating other samples or anything like that.
There you go. I can open more if you want.
Oh, that's fine.
Oh no.
So now we have the question of, do we want to do number 5 at the same depth, given that 4 failed?
I think we do.
Yeah, I think we probably do.
Yeah, I agree.
First, we have to get rid of ball 4.
That's true.
It's
Might be
like
there
awkward
forever
to make
now.
the wrong size for this.
It's like going to be the trickiest sample of the day.
Let me know if you want the box open more.
No.
There we go.
Oh, nicely done.
Thanks.
Oh, wrong way. Sorry.
Bummer. Okay, you guys want 5?
Yes, please.
Yes,
Okay.
please.
Something that wasn't even one that was that sticky when we were routing.
Just hold on 1 second.
Yeah.
Santiago, the only thing is we're not getting a CO2 peak at 680 meters. Do we want to go slightly higher? Or just take it here, because that's where we saw ORP?
I think we'll just take it.
Yeah, okay. I am also fine with that.
What do you see?
Yeah,
It's like
thanks.
jellyfish.
Oh,
I thought it was a squid for a moment.
all right. I am watching.
What the heck?
Oh, it's not going either. Are the last three connected? They're sitting
I mean-
very far out. Like, much farther out than
Yeah.
they normally do.
They should be.
Like,
Yeah, no,
compared
I'm
to
with
the
you.
other three, right?
Hmm.
Like,
And the bottles are definitely cocked at their end, right? It's just whether the lanyards are connected all the way through.
We could try 6.
Mm. But all three are on the same swing arm, right?
Okay,
Should we try 6 anyway?
I'm gonna try 6.
Just for yucks, but
Or if 6 has,
if 6
Ryan and I
doesn't...
are sort of coming to the conclusion that they may not have been hooked
Right,
up.
that whole set of 3.
Like the first 3 were hooked up, are always
Right.
hooked up.
Yeah, do you have to unhook 4, 5, and 6 to get the swing arm open?
Yeah.
Okay, so it may just been in the...
Yeah. Did we have to get the swing arm open in order to do the thrusters? Probably.
They did. They
Yeah.
had both gates open. Yeah.
So that may just be it. They just didn't rehook it when they put it back in the water.
Did you want me to try 6?
Yeah,
Okay.
please.
Yeah, this is really long and dangly. Nope.
No. Okay, thank you
Nothing
for trying.
doing.
Nope.
Rats.
Okay. Let's secure everything and head off for the next waypoint.
Okay.
Can we secure lasers just for a little while as we take off?
Yeah.
I think let's make some pretty little footage for a couple of minutes with these ripples and the mushroom corals.
Santiago, with the eDNA sampler, the pumps have very different flow rates. So one of them is pumping twice as fast as the other. Do you know if you would prefer to have it that I let one overshoot and then just have it so they both sample the same
little
section of
fish.
seafloor? Or whether I should do the, like, as soon as it gets to target volume, start another one, but then we'll have them all, like, laddered rather than at the same point for the
No fish.
duplicates.
I think it makes most sense to just do it by
There
what
was
we
a
see on the
fish.
seafloor.
One sec. I'm
I wanted to look
gonna-
at it.
Can you say that again?
Can you hear me?
I can, but really quiet.
How about now?
Yes.
I was saying that I think it makes the most sense to make the call on when to stop based on what we see in the seafloor.
Okay.
So you've been running them, I presume, while we've been seeing this kind of anthomastus gardens and
Sorry,
This-
Megan, if
this
you see
kind of-
something, just
Yeah, yeah.
grab my attention.
So
really.
they've been,
I
the
didn't wanna
target was originally just
over-talk
run them
the
through
background.
till 120 liters and then stop, but we could do it by feature instead if that's what you prefer.
yeah, how much volume do they have?
The first two both had 120 liters, and then this next two, one of them's already on
our help.
105,
Headset's all
and
the
the
way
other
up.
one's only on 57. So there's a bit of a difference of flow rate there.
Yeah, large difference. I would say keep pumping. Yeah.
Okay.
I mean, I was just thinking what would be a good cue to make the switch, but you know, I mean, the idea is to capture the differences in communities that we see with depth on the seafloor, and so
If you see anything here you want to zoom on, let me know.
I will shout.
How many pumps do we have left after the ones that are running now?
We have 15 in total because it's the
And
shallow
how many
depth.
are we in? How many do we have? Have you taken
We've
so
done 2 have finished and 2 are currently running.
Oh, okay.
So we have another 11 to go.
I would keep these ones running then, because then if we do suddenly see something to do something different, like
yeah.
our community, we've got plenty to reserve. So
I think Annette's main concern was just timing of the dive. It's only 6
Just
hours
drop off.
Mhm.
Anyway.
left.
Let's
And
go look
that
over the edge.
Yeah.
but we're
currently,
going to
So
do a
we'll
whole
take-
second transect. So
yeah, but I think with already they take about
Oh, that's a long way down.
It's like in Finding Nemo.
1 hour 20 to pump through with the current volume we're using.
Mm.
And it'd be a shame to come up with empty pumps.
Yeah, I have a little bit of different philosophy about that.
Okay.
Because what matters is a good sample, not
Yeah.
many samples.
Yeah.
But let's say for now, let's keep it running, and then maybe in
if we keep these ones running till from here to the summit at the next waypoint,
yeah.
if they haven't filled by then that might be a good reason to switch.
Yeah.
When we're doing the descent to wherever we go next for the second transect.
Okay.
Yeah, I agree.
Yeah.
We could do like a water column. Oh yeah, that's a point. Santiago, do you have a preference for which of the available sections we do for the second uphill?
Good question. Let me see the dive plan.
Bridge nav.
We had the options.
We'd
Do
like
you
to
have a copy of the
move
dive
20
plan?
meters 157.
So we have that one or that one. I guess we can only go to 1,000 meters, whichever one we go. Okay. Where we saw the chemical anomalies, yeah.
Have you looked at these little
Yep.
black things
So
yet?
go and then do
We
the
looked
ridge.
at one earlier.
You like the look of that ridge? Yeah.
It might be like a
And see how
xenophile
far we
for.
get. Yeah.
Go for zoom video?
Okay. Sounds good.
Which one did Santiago have preference for?
We're going to go to 1J and
That's
then
the
come
only
up the
thing I can
ridge
think it could
They
be.
kind of look
and
slightly
reserve the option to cut
biological
across to
anyway.
the vent
Yeah, it's really
at some point as we
organically
come
shaped,
shallower.
so it might be a giant protist
Okay,
called a xenophile 4.
Okay.
go wide video.
but I've never seen them that color before.
Hmm.
But it could be because
So
there's
maybe
like
go
this black
1J to
sediment.
3J and then
Yeah.
3J
They
to
like
4H.
build these little homes.
What's the reason
Oh,
that
there's a fish
you
for you.
preferred
Yeah,
the
that's another one
1J
of those
route
green
rather than the
eye
1H route?
fish.
I
Either
think
of the two of you.
1J is along a
Both.
rocky ridge, so it's just like
Go
a,
for Zoom video?
a happier,
Go for Zoom.
happier place for corals and things.
Yeah, I was thinking maybe we can do that and then move along
Yeah.
isobath.
Megan,
Yeah.
how do you get these
So
zooms?
go 1J to 3J and then cut across
That was,
from
Oh, I
3J to
yeah.
can't
4H.
the seafloor,
and
that's
that
why.
we still get kind of that
Go wide,
coverage
Go
of
wide.
the potential seafloor, sorry,
Actually, I got
CO2
it.
spillover,
So.
Yeah.
Yeah, I think there's a
and then maybe check out the potential source of that water column anomaly
oh,
that you
there's
saw with
another
Sentry.
fish, a wiggly one on the left side.
Oh,
Actually, that's the nice thing. If you do go from
squirrely
3J to
Yeah.
4H, then because you're on the same isobath,
Mm-hmm.
So
then
if
that
you
might be
come
an environmental conditions, not
down
change
a
in
little
pressure
bit.
or anything else.
Right.
But it would be heading towards a plume. So I don't know
Right.
if we'd be able
Go
to
for Zoom
test
video?
the
Zoom it in?
spillover
Well, we can go
theory
uphill
so
from
well.
4H because that's still downhill, so we
so
could then
cute,
from
it's a little
4H we
reptile.
could continue uphill
Okay, go wide video. Go wide.
from there towards the
Does that make sense to you, Mary?
I think it definitely makes sense.
or did you want to go
From,
to 3H and then go from
So
3H up to
he's looking for
4H?
some breakfast.
So I think it depends on what we want to
Around.
see.
Little
So
fish on
on
a
the
mission.
current transect from 1G up, we didn't really see any CO2 at all. So not having, until recently, until this last
Mhm.
waypoint where we got some small features. If we want to compare the magnitude of those features on the east face or the northeast face,
Where was the
then-
CO2 plume? Was that between 6G and 7G?
so that was at
It must be because where did we find Niskins? What were the sample numbers for the Niskins?
yeah, 1 second.
Niskins were number 1 was 247.
Mm-hmm.
Number 2 was 249. And
So
number 3 was 251.
the plume was at 740 meters, which is
There's
So if you look at
another
those
fish.
contours, that's between 6G
Yeah.
and 7G. So we could go, if we went 1J, 2J, 3J, and then we could jump down from
It might be a
3J to
reptile
3H.
too, like the one that we were just looking at.
That would get us back. We'd go from 3H to 4H, would take us back through the depth range of the Niskins again.
yeah. I wasn't so concerned about
Zoom
the
video?
depth range.
Go
I
for
think
Zoom.
the
Well, we'd like to come from below up through the depth and then
no, I
Sorry,
follow
go
that.
wide video.
I
Go
was
wide.
more thinking
I got too much going on.
whether that any signal approaching from that direction will just be dominated by the fact that we're approaching right next to a plume rather than getting the more kind of overall trends of how the emissions move away from the sources. I think probably with the time that's
Bridge
still a
nav.
good option to do the 1J come up, but I'm not sure that we'll be
Yeah,
able
we'd
to
like to
gain
have
much
20
information
meters,
by
157.
yeah, we may not be able to gain very much just because we're going to be approaching so close to a plume. It may be worth going slightly above the 680 on that J transect and then
Go
go,
for Zoom
you know,
video?
even just
Go
10
for Zoom?
meters above that
Mhm.
isobath and then drop down again and move across
Well, it's got a little,
just so we
this
can
is
probe the
Malacocephalus.
water column structure there a little bit before we start approaching the
Got
plume directly.
dusky fin. Very
Um,
cute.
Okay.
I think that would maybe fill in some of that gap in
This is a
information
different
about how
reptile from the
the
one we were first looking at.
buoyancy of the gases
All right, go wide video.
affects
Go wide.
their spread. But let's see how we're doing with timing when we get round there, because I am very aware it's a pretty short dive and there's a lot to see and a lot to do.
Mm-hmm.
what you say makes sense to me too, so
That sounds good.
hopefully we can cover those, all those.
Cool. That sounds great.
Oh, shrimp.
Just looks like a bunch of broken Doritos.
Oh, can we
Ooh,
Yeah,
let's
you
zoom in
wanna
on
That
this,
looks like a
please?
map.
Can we?
Everybody's like,
Let's hang out there.
all right.
so say Mary, this is a good cue to change
Is
bombs.
this a potential place where you want to
I could do it if you want.
poke
No,
around
it's
for a
good, it's
while,
good.
Chris?
Yeah. Yes.
Okay,
Okay.
Stop
So
the
let's
ship.
stop the
Ship
ship.
move.
Yeah.
Bridge nav.
I'll sit down right here.
Yeah. I'm sure there's
They like to
potential
hold
to
position.
like temperature probe.
Yeah, yeah,
and-
yeah. I'll just sit down right in the...
Here's the dusting for my Dorito chips.
So, data, I've just stopped pumps 2 and 3, and I'm about to start pumps 4 and 5.
Okay.
All right.
So do you think this is bacterial, Matt?
Yep. I think it probably is. If we can sit down and have a look.
Yeah, we are sitting down.
Do you remember we saw like the spider's web microbes? I think
Yeah.
that might be what's hanging out on the
Alright.
underside of that basalt just
We're
in
sitting
there.
down. I can get closer if we need to. Do you want to zoom?
Yes, please.
All right, go for zoom video.
Going for zoom.
Yeah, this is what
That
we were
looks like
seeing
it would have
Yeah.
a really weird mouthfeel.
around the
And then
northeast.
video,
Yeah,
the more
that
you
would
can
be
wander around and look at little places, but I think we'll have multiple views of any of this yellow stuff. We're particularly interested in like the whitish gelatinous stuff. That's the freshest stuff, so
Yeah. Was there a spot up there that was...
we're still looking. There are multiple spots at the moment, but I wanted to
Do you
look
care
around.
if I pan around?
No, it'd be great.
Okay.
Panning around, but then stopping so we can just see if there's any
Yeah.
evidence for any shimmer anywhere as well.
Okay.
Mary, do
I
you got?
haven't seen any of that yet, but
There's some of this.
can we pan down a little bit
Yeah.
on that hatch? Yeah.
Like that.
Can we sharpen that at all, video, or is it just too wafty?
It's
No,
very
you got
wafty
the-
right now,
Okay.
but it's as
Times
focused on that
for-
yellow as I can.
Yep.
Pump 2 and
If we
Yeah.
can,
So pumps
okay,
2
if we can pan
and 3 both
right again,
finished at
please, Rye, and then go
12:55.
up underneath the, there's a little overhang in the rocks.
Like,
So if we go
more?
straight up from
the
there,
depth
sorry,
is
Oh,
go
sorry,
658.
yeah.
back there, yeah, then if, I think if you go up now,
Yeah, you're talking about that spot,
Yeah.
right? Yeah, let's take a look
That's a
there.
nice shot on the cinema cam.
can we zoom on this, just to,
This is full zoom for you.
Here, I'll tilt up a bit.
and what are the volumes for those?
So for pump 2, that was 127,
is that full zoom, or can we go
102.
any
This
further?
is full zoom.
Okay. Yeah, I think poking some of this with the temperature probe
Okay.
wouldn't be a stupid idea. I'm not sure how thick it's going to be, but
Okay. Sounds great. Would love to.
Pump 3?
Pump
Alright,
3 was
go wide,
70072.
Going wide.
70070.
72 at the end.
Chris, do you think
Is
that
this going to be
Yeah.
close enough for the temp
Sorry.
probe, or you want
We
me to
should
go
go-
closer?
And the start
take
times for
photo
Would be
button
nice to
on the
pick up,
cinema cam will?
move a
5
little
and
bit.
sorry, Santiago, you're very quiet. Could
Both
you-
also started
I
at
was just wondering if you
12:55
think that
and both
the-
at 658.
take photo button
Sorry.
on the cinema cam will take a grab of that.
Oh.
It looks like it's a really nice
yeah,
shot
we,
there.
we've been told
Oh,
not
there.
to take pictures on the cinema cam anymore, but
All right, a little to the right and
maybe we can
up.
try.
like
you
I'm
can
getting
take the
good
pictures, there's no
good data on
guarantee
the
that
temp
you'll
probe
get them in the end.
in spite of a ground fault on it, so we'll use it.
Yep. How are we doing for chambers on the slurp?
We have 6 chambers open.
And when we slurped microbes before, it did actually retain sample, right?
Yes.
It wasn't a stupid thing to do.
No, it worked really well.
Okay.
How's there?
That's great.
So I think, Santiago, I'd suggest we try and take
Beautiful.
a sample of the microbes as well here to compare with the stuff inside the crater.
Yeah, sure. It looks very similar.
I am set down.
Okay.
It looks like
So
a...
I can see the temp probe?
Yeah, absolutely.
What's the ambient temperature here?
6.
6.
I'm gonna retract
I
a
suspect there's not going to be a lot of change in temperature, but
No, maybe 7.
maybe not.
Oh, that's the wrong thing.
bit. Oh yeah.
And we're getting some increase in CO2. Not
Oh, you
very
are?
Oh,
much,
good.
but 10 ppm maybe
Mhm.
since we moved this location.
So Chris, I don't think I've
Leo,
shown
can
you
you give me,
these
like, a little
plots
bit
that I
more
made last
iris?
night from the mapper
Yeah,
data
totally.
from Herc.
Somehow I
The
have to grab this without grabbing any of the
we
That's
saw
all.
like 4 or 5 different plume signals during the ascent from the previous dive.
Do you remember how big
Oh my
the
God.
signals were in millivolts? Are they?
You don't like your silly
can show
straw
you.
temp probe?
I think they were
What's that?
You don't like your silly straw temp probe?
0.2?
0.2.
No.
Yeah, they were not very
I
high,
didn't do
but
as good as I should
0.2
have.
volts?
millivolts.
I
Is it
don't
actually cable
remember.
wrapped around the probe?
Millivolts, I think was your scale.
Yeah.
The difference
I
is,
think
yeah.
that's
It should
very
be-
silly, and I think I can probably fix it if I go the right way
If
here.
it's 0.2 volts and
What
200
is
millivolts, that's a
happening?
good signal.
Oh, it's
That's
got
kind of
one
what we were seeing with
around
Sentry.
Yeah, it sure
Yeah.
does.
It was about the same.
Yeah.
Oh yeah, those are
If you
very
stick it
healthy
to
size
the
signals.
thing and
Yeah,
maybe
and it was
try to-
The rule
good
of
over
thumb
that.
is anything above 5
See, like, how
millivolts
does
is
that even
significant.
happen?
So
I think you
200
gotta go
is
the other way.
significant.
And it was correlated with a nice CO2 feature as well
Yeah. Mhm.
for at least one of the
And then it'll
spikes, one of the ORP hits.
sort of pop out. But you gotta go all the way.
Ah, this is the first derivative, sorry. I don't carry that around my head. It was, but yeah,
Nice. Beautiful.
Yep.
All right.
Good teamwork. Thanks, Rye, for having that
Yep.
figured out.
so we're
Here,
at 660, so
Okay,
it's very
let's
much
pan
this
up
one
so I
here,
can
right?
see the map that we're looking at
Nice. Okay, let's see what happens with the temperature probe. Thank
Temp
you.
probe is on. It's giving good. Is it giving good data?
Here, let's see.
And Megan, did you get those last 2?
I did
Yeah.
not.
Okay.
kinda matches
no,
the-
it's
254
last time
and
it gave data
255.
was
Okay.
Oh,
No, it's good.
oh, is it on?
it's on. And last time it gave data
I'm seeing
was
data on Grafana.
but the last data I'm getting from it was like 4 minutes ago.
I've got it shown the last 5 minutes. I think it came good again.
Yeah, you're seeing.
Or is it just not
I have-
updating,
The
it's
last
just
data I
rewriting
saw
the
from
same
it was
data point?
4 minutes ago.
Did we pick it up 4 minutes ago?
No, I don't think so.
Do you want to power cycle it?
Yes, that is exactly what I was going to do.
Give it, I don't know.
Was it 4 minutes ago when I said we should see how thick the sediment was using the temperature
Probably.
probe? It heard me.
Yeah.
What's the
I
magic?
mean,
Don't
it could
do
be
that.
that I picked it up, but if that's the case, it's being very fussy about
Mhm.
things.
All right.
And that temperature probe
Turn
would
it back
never
on.
do that.
It has a pretty good strain relief on it right now.
There you
I'm
go.
not- there we go. Okay, great.
Yeah, and that's
I'm
good.
not I'm not super impressed with it if it's complaining about the current state of things. It's got all the strain relief it could ever want. Okay, Chris, if you want to circle what you're interested in looking at.
Yeah, can we go half zoom? But I think I recall that there was white gelatinous stuff in this mix here.
Okay, I'm going to come in for a half zoom.
Thank you.
Yeah, I can told too, so.
Yeah, if we could get the tip in that kind of area just there,
Okay.
please.
Poke the goop.
Oh yeah.
And the
you
temperature
bet.
has been going up just while you've been holding it. I mean, by tiny amounts, but... Ooh. That's got
That
some
is more
depth to it.
gelatinous than expected.
Oh.
Whoa. What is happening?
It's eating the temp probe.
Yeah. That's very funny.
It's gonna eat the whole ROV.
Oh, that's going up. Let's
Is it?
hold that there. On Grafana. I mean, it's
Wasn't
like
the
it's
ambient
tenths of a
6?
degree Celsius, but it was at 6.2 and it turned a corner
Yeah,
when
it's
you
going
stuck
up
it in
a
the
little
probe.
bit.
I can probably go deeper.
No, just hold it
Okay.
there because it's still doing its thing.
I didn't stop for any reason other than I was nervous that maybe the microbial mat was
You might
eating
just
the
swallow
temp
you.
probe.
Yeah.
Yeah.
It's like when it gets halfway up, when it gets to the
Oh,
elbow,
7.
Then maybe we'll
it starts.
get
yeah,
out of here.
Yeah. We'll just cut the feed.
How are
My-
we from a time and drinking bird perspective?
Yeah. It's beginning to level off, but it's between 7 and 7.5.
So does this qualify as a cold seep?
I think it qualifies as something. It's some kind of low temperature flow, I think. Must be. This is the range of not exciting temperature range, you might think, that we found in the 100-day-old lavas off Kilauea in 2018. And they had different chemistries and completely new microbial species in the stuff
Oh,
that was
wow.
yellow and looked like microbial mat like you see everywhere else. It turned out it wasn't the same microbes at all. So I think we have leveled off. Chris, would you agree?
Looks good to me.
Looks like,
So it's 7.3.
yep. So would you like to do drinking bird before we slurp, Mary?
I walked at the wrong time. What did you say?
I was saying, would you like to do drinking bird here before we slurp some of this stuff? Because I think we definitely want to take some microbes home.
Yeah, let's just do a quick drinking bird. I think we'll
Shall
see
we-
pretty quick if it's going to increase. And
Actually,
then
let's just see if there's anywhere a bit warmer as well, since we're
Okay.
here. Let's take the temperature probe out from there. Thank you.
Like maybe that crack. Whoa.
Yeah.
That's like so gelatinous. Like the
So
crack
let's-
above here.
Yeah, if you just hold it there a moment, I'd just like to see the temperature go back down again, give it a chance to
Okay.
get back to... Actually, before you do the crack, let's try over here as well.
Okay.
The crack has less of the pale stuff and more dark brown, so I think that might be less active than it...
Okay.
Okay, temperature probe's going down, so I'm ready for you to
Okay.
go somewhere else, please.
right behind the temperature probe cable is a white crack, which could be another one to try.
Mhm.
So right here-ish,
Yes. Can you, like, rotate so it's more vertical?
the temp probe?
Yeah.
Yeah.
Temp probe's
Sorry,
going to
I'm
think
gonna
I'm
try
its
and
nemesis.
manage the
It's like, oh, not that guy again.
cable a little bit here.
It's not enough just rubbing my face in the dirt, but he just wants to face plant me.
Just due to the length of the elastic, no is the answer to
Okay,
your question.
all right. Okay.
Wait a second, I'm going to do a little bit of
Whatever
maintenance
angle we
here
can
on
get.
the ridiculous angle that we have right now. Can you go wide video?
Nice.
That's always an exciting move.
Is dropping something?
Trying on purpose to drop it and catch it before it drops. Okay.
It was very impressive.
Yes, we got from 7.3 down to 6.0, so it's more than 1 degree above ambient.
Okay,
Inside
so
where we were before. Yep, back
We'll
in
try
that little
here?
bit just there, where the
Okay.
palest yellow is, please. There we go. That's lovely.
Do you want zoom on video?
that is as far as in as that's going to go.
Great. If we just hold there, please.
Look at the lower view. It's like,
Funnily enough, the temperature probe is looking spectacularly unimpressed by that piece of that patch.
Can you zoom in a bit, video? I want to
Yeah.
see what's going on with the cable here.
Totally.
Yeah, no rise significantly yet.
It looks fine.
it looks fine.
Yeah.
Mhm.
It looks like
No,
it
it's
just wants to be wrapped that way.
It's just slowly beginning to rise now.
Okay.
But in the other place, it kind of like went asymptotic.
Yeah, it went up a lot quicker at the other one.
Yeah.
Okay, let's...
Data, I cannot hear you.
Data, I think
wonder
your
if
mic
that's-
is just really far away from your face.
Oh,
There you
is that
go.
better?
That's a
Yeah,
lot
that's better.
Okay.
Great.
So that's not nearly as impressive as the other place, Gabby. So let's try up in here somewhere, please.
Okay.
So far, the place we started was the winner. Everybody bet now, higher or lower.
I'm going lower.
Yeah.
Yeah. But I think this
Might be-
area might be one to try
Okay.
if that doesn't.
Rats. I'm losing my grip here.
I can always get closer if you want.
I think we need to for that spot.
Yeah, all right.
I'm
Get-
just gonna bring this in so we don't whack it.
Yeah, go wide video.
Actually, before you move, let's do
Mhm.
this spot. Which this can you, Mary? Can you guide me to where
thinking
you were
that bit,
interested in?
Mary?
Yeah, the really pale area.
Okay.
The pale microbial area.
Just to the right of the lump.
It might be a bit rocky, I'm not sure.
It's looking pretty slurpy.
It's pretty gelatinous here.
Mm-hmm.
Okay.
Whoa.
It's quicksand.
Let's hold that.
It's gone.
Too
Yeah.
bad Adam doesn't want to push core here.
Yeah.
Does the
Actually, that
knots.
could be a much better way to do it than to
Slurp.
slurp.
Can you
Interesting.
process
You
the
won't
microbes,
get
or it's
as
harder
much.
to...
I think the slurp will give you more of the mat.
Right,
The push core will
and then the
probably
push cores
mix
are
up-
hard to deal with. Temperatures, oh, this is good for temperature. Gone up above 6.8.
So is this Price is Right rules? or-
Passing 7. I think it is.
I hate Price is Right rules.
I don't like them
I
either.
think closest wins, whether you're above or below.
I
Because
agree.
if you're playing Price is Right, the game is like sniping it from your neighbor.
But
you just like guess as close to your neighbor as possible.
But we only asked for higher or lower. We didn't ask anyone to bet on temperature.
That's
perfect.
true.
So it's
And I was
already
betting
too late.
lower, Yeah.
Yeah, that's perfect. Higher or lower is great. Over under is great. I'm not on board with Price is Right rules.
No, I'm with you. I don't like
I
it either.
feel like you can act in bad faith. What's going on?
You give me my coffee?
I'm gonna
Sorry.
confess to complete ignorance soon and just say I have no idea what Price is Right rules are.
Okay, that's great.
I
Ah,
also don't know what
nice.
they
Because
are.
I have used Price is Right rules to beat Rennie, and like been like just 1 meter off of a guess of his, like, and I felt bad for days. And so I'm like, this can't be good.
Days.
So the Price is Right rules are if you get the closest to it without going over. So if you
Oh,
go
I
over,
see.
it doesn't count, even if you are like 0.1 over.
So if somebody makes a guess you think is good, you just guess higher than them
Yeah, by like 0.1.
by 1,
I
and
don't
then
like
you
those
take their
rules.
guess from them.
Yeah. I could do
It's
that.
evil,
No, it doesn't seem right.
and I have used it for nefarious ends,
Mhm.
and I still obviously have not made my peace with it, so I'm done.
So this, if
This-
anything, is a little warmer than we were before, so I don't think we should worry about the other crack higher up. I think
Okay.
this is already a perfectly
Yeah.
good place.
Okay,
Yeah, really
That's kind of
7.8
7.
right now.
and so are you interested in slurping the mat, or?
We would like to do a drinking bird deployment
Okay.
first
Yeah.
of
Okay.
the undisturbed system, or relatively undisturbed
And
system.
Mary, have you seen them do a drinking bird deployment yet?
Yes, I have.
Okay, so maybe you could guide me through it.
Sure. So the only time I saw them do it, the the bungee fell off and they
Okay.
forced it down with a with the arm.
All right.
So there
'Cause
is now
I've
on
seen
it
I've seen
a-
Tito do it.
Okay.
Yeah,
do
I
more
think
you
that was
want to
the
put
recently,
the
same
temp
dive.
probe away?
Maybe.
No, I think
They-
that was the
It's kind of level up.
photogrammetry dive.
It's kind of
Ah.
level
Can
up.
I
except
put the
one.
temp probe away?
You can. Thank
Okay,
you.
great.
There is now a little ring of cord on it that
Okay.
can be used, but I don't know if that's useful or not.
Yes, we got to 7.8 there, Chris. Yeah.
Yep, I got 7.81.
Well, glad I didn't say 7.82 then. Now that I know what Price is Right rules are.
Yeah,
Yeah.
and now that you know that I can't help myself but make evil bets on Price is Right rules. I'm not a good person.
Price is Right is huge in American Samoa.
Is it really?
Yeah.
Yeah, because it's the only thing that's on TV.
Oh, it's the TV show.
Yeah, during the day it's Price is Right, and in the evening, like late at night, is Days of Our Lives.
Oh boy.
Okay, so drinking bird. Can you retract all the way and look over at drinking bird?
Yeah.
So have you done any touch-ups on drinking bird, Samantha?
I haven't, no.
Oh. Here we are then. Here's the original form.
Okay. This potentially involves 2 arms. We'll see how available this loop is. Because this is a much higher up loop position, and I don't know that it's a great this arm grab. I might need a bubble view.
Yeah, do you want to?
I would love help with bubble.
So based on one data point, I would love to hypothesize a causative relationship between the high abundance of mushroom corals and these microbial mats.
Oh, that's...
Sorry.
Cool. Oh, that's a
Yeah,
great view.
hopefully there's a correlation. That'd be kind of cool.
And it's always just like when you see a bunch of stuff, then you're in places where you haven't seen a bunch of stuff before. It's like, what's going on there then? What's
Yep.
special about that place?
Yeah, what are they drawn to, or what's
Mhm.
encouraging their growth?
Let's see if I can prove myself wrong before we finish the dive. We're like, nope, that was just nonsense. That was just a really nice color palette of cream microbial mats and
Okay.
bright pink.
Don't do that. Huh. Okay. Well.
Here it comes.
It's one way to do it.
Yeah, sure is a way to do it. It is the way that I did it.
important part is you did it.
What's that?
The important part is you did it.
It's the way you thought you did it.
Yeah.
I jinxed it, sorry.
I didn't realize it would have to go down so far. I guess we have to hold it in place?
I think it locks at the bottom, but only once it's fully drawn out. And
What's that?
basically, it does lock once it's fully horizontal.
It's that over center, but I thought we had closer access to it.
We could just leave it with the arm for safety.
It'll
What's
be
that?
fine.
And then we could leave it being held with the arm at the bottom rather than try to
Oh,
let
I see,
go.
rather than get it locked in place.
Oh, no, yeah. I guess the arm doesn't really do any harm being there still. I love the googly eyes.
Mm-hmm.
My only contribution.
Because
to the
is that from your
drinking bird.
collection? Yeah.
It's my legacy.
Yep. Well, I heard they're getting in short supply.
We're finding them in various places.
I feel like we missed an opportunity. We should have asked
Oh, nice.
Justin to
That's
dress
better.
Sentry up as drinking bird.
Oh, yeah, there's still time. I don't know if he'll have time.
See
but-
your angle that you have?
Yes,
in
I see it.
level? Yeah, okay.
Oh,
What was the ooh?
that was a good catch. As in, like, I don't know if you let go deliberately then and it
Yeah.
stayed, or if it just fell off and stayed.
Well, let's go with yes.
Okay.
And now would you like to gently relocate the front of the porch so that the nozzle is right over the fluffy stuff?
That would be great, right over that pale area where
Okay,
we...
I'm going to move the arm up a little
we
bit
wanna
so
go
there's
to
room to push
kinda there.
drinking bird.
It was right behind, right? I think it was right behind where the arm is.
So
Actually,
there's
yeah.
there's real risk of getting sediment in the schnozzle here if we do that.
I
I do have
am
an idea.
happy to take the risk. We
I
have two
have an
filters,
idea.
so
Mhm.
I'm just going to grab the drinking bird and pull it up a little bit, and that way we can move. Okay, drinking bird is mine.
amazing.
Okay, you guys ready to...
So I think if you're looking up at Gabby, or right,
Yeah.
you can see where we want to. We'd want the front of the drinking bird to be right over those pale yellow patches somewhere.
Yeah.
I think it was where
A
we
little bit to the
sampled
right.
was more like
Yeah,
under where the
they
arm
were the
is
same
currently.
to within like 0.3 of a degree, so I think within the precision of landing,
Yeah.
anywhere there when a little forward would be good.
Like right
Just
here?
like there, yeah, that looks
Yeah,
perfect,
okay.
thanks.
Just try to stay like that as I come down.
Okay, good.
Yeah, we are down there.
If you look at that one, then it's vertically above, over to the right. It's closer to where you... Seeing a little nudge?
Are we good, sides?
Might be going up a bit, huh?
I think still
Give it a couple of minutes to
Yeah.
know what's...
it's a bit,
I'm going to put it back down.
Yep.
For
that sounds great. Yeah, I'm getting
You're-
some CO2 signal
yeah.
coming from
Huge
this
odds.
region. Is it possible to back up maybe 10 or 15 or 20 centimeters with the vehicle, just so it's not in the rock?
Yes.
Thank you.
Am I good to try to do that, or do you want to?
I'm going to put the jaws over it just so it doesn't fly up in the air. Okay, go for it.
So
Too much?
Crispin has just asked online what the device being used is. It's the inlet for the SAGE gas sensors. So there's the 2 gas sensors which have been
There we got a little.
got their inlets tied together. So we'll sample both CO2 and methane in this same region right above the microbial mat.
Go down, please.
Yeah, definite CO2 signal that we're getting here. Really, really strong. Yeah, that's perfect. If you can
Is it?
stay, that's so good. It looks like from the upper that it's right over that white region, which
Okay.
is where we want it to be. So that's pretty good.
Let me see if I can get it. Okay. You're stable there.
Thank you, Ray. Nice flying.
No worries.
And Chris, we don't know if we found methane in any of our samples yet because the gas-tight samplers are actually sealed up and they're heading back to Lehigh, which is where they're going to get analyzed. So
Okay.
Jill McDermott has the fluids, so we're going to know that.
All right.
So we did actually find methane in some of the vent samples in SAGE from right next to the bubble vent. But
Oh, okay.
yeah, a very small methane signal that was associated with the really strong...
The much higher CO2 signals.
Yeah, but slightly unusual maybe. So I would say it's a provisional trace methane rather than real trace methane until we see the IGT data.
So maybe that is just a little bit of reduction of some of the CO2, for example.
Yeah, I think
It's like an impurity in the main gas is the CO2.
yeah.
Or you think it could be some other peaks that...
No, I do think it's a methane signal, probably, but I think there's a lot of, it's hard to be sure whether it's just a sort of bacterial origin from sediment, from microbial activity in the sediment that was stirred up while we were sampling compared to one that was actual venting fluid. So I think it will be see how IGT data compares to the methane signal that we detected.
And Chris, you're right, this is not an ultramafic rocks area. This is very much a basaltic volcanic system. And so traditionally what one finds here is a lot of magmatic CO2 coming out. And that's entirely consistent with what Mary's been seeing in the SAGE sensors. And the gases, it's the same kind of thing people have reported from the seamounts around Hawaii as well, the Kamaehua Kanaloa seamount, formerly known as Loihi, has very similar things, but although there you don't see the free gas bubbling out.
So I think we're sort of approaching a stable value with SAGE, so we could probably call it a day.
Happy with that?
This. Yeah, I think it's sort of
Nice data
settled
set,
out at a specific value, and yeah.
How far did you get up to?
So the peak was 375, but I think that might have been from inside the rock, basically, and not necessarily the same as where we are right now, and where we are right now is settling out about 350.
Okay. Did you want to try another little move 20 centimeters to the right, or are you...
I think, just in interests of timing for this dive, we have a lot of objectives
Okay.
we'd like to achieve.
Okay, let's stow the drinking bird
yeah.
then, and we'll take a slurp sample, please.
That
That was
sounds
very
good.
restrained of you, Mary.
I know, much though I love the drinking birds, I also really want to see if there's a new vent. So, you know, there's definitely
Yeah.
benefits to...
We'll get our voucher sample from here and keep time in the bag for even better discoveries. If the worst thing that happens is we found a new microbial site with active fluid flow on the outside of the volcano, that's a pretty cool discovery already.
Yeah, yeah. I mean, it's definitely got a temperature anomaly associated with it.
Yep.
And definitely a CO2 anomaly associated with it.
I do have a question.
And we'll probably be here long enough to get a good DVL reset at the end as well and have really good navigation and know exactly where it is. Sorry, Belle.
Does higher temperature indicate more biological activity or a different kind of biological activity?
Tell me that one again. Sorry, my brain is slow to ingest tonight.
No worries. Does higher temperature indicate more biological activity or a different kind of biological activity?
Ah, yeah. So that warmer temperature suggests that there's some water flowing through the rocks deeper down and coming from deeper. So that inside the rocks, there's a thing called a geothermal gradient. So that suggests water's coming up from below, which at some warmer temperature is probably dissolving up some interesting chemicals from the rocks and bringing them up to the surface. So then that might then change what kind of metabolisms are available for the microbes. So yeah, it becomes a different kind of biological activity.
It's definitely not sitting on the magnet.
So this pale yellow color
Yeah,
I think
it's
I got
the
was
magnet.
Yeah.
something we were seeing a lot all around the Napanua cone was alive with this kind of microbial activity. We haven't seen it on this dive until
Okay,
now,
and
so
we're
it's
suction sampling
distinctive.
now?
Yeah.
So it's definitely different, but if you look at all the mushroom corals around it, there seems to be a lot more life in the corals. But then the question becomes, are the corals finding more as filter feeders to do if there's these extra microbes? It's a different flavor thing on the menu for the corals, maybe. That because I think we're about to see that it's quite soft and fluffy stuff that could get lofted quite easily.
Did we put something in 2?
No, 2 should be empty.
Okay, there is definitely a little bit of something in 2.
Okay.
That isn't where we put the mushroom coral before?
No, those are all in 1.
Yeah, and we never passed over to 2.
Ah.
Sorry, what was that, Megan?
There's just an amphipod that swam up in there.
Yeah. And then another little something in
It's
there.
like a little bit of
did you get a flush
mushroom
on it?
coral.
Yeah, I did get a flush.
Okay, do we want to put this in 2? I think 2 has the small screen, so it seems like a good candidate.
Oh, yeah. Yeah, that's a
Okay.
good reason. Good call.
Can I have some suction?
Absolutely.
And a half zoom video.
Have Sam.
I'm going to turn suction down, though, so that you get... How's that?
That's excellent. How do we feel about that sample?
I think that looks pretty good. The chamber looks pretty happy, doesn't it?
Yeah.
Do you need any more?
A lot of stuff in there.
I think
Hopefully that should be good.
that should be enough for you to
Yeah.
cope with, Chris. To
Okay.
Great.
That should be
Go
enough to
wide.
get some stuff.
Go
in
wide. Okay, is that it?
I think so.
Yeah, I think that's good for me
That's
as
a
well.
nice little data set. And
Yeah,
That's great. So glad.
yeah, really, really nice. Thank you for the manipulating.
Okay.
Okay. You ready to keep going?
Actually,
Yeah.
could you do one thing before? Is just zoom in on the area where you removed the mat and just check if we can see any fluid flowing.
Yeah, sure.
It should be pretty speedy.
All right,
And
go
Megan, that
zoom
was
video.
256.
And I'll get us nice and lined up so we can
Okay.
take a good look.
I don't see anything.
Megan, did you copy that
Yeah.
at 256? That's,
yeah, 256.
Yeah, I don't see anything.
I think you wrote it
Oh,
in
I wrote
156.
156.
Yeah.
And
I don't know why I did that.
Okay.
'Cause
but that's
it's
consistent
2:30 in
with
morning.
the stuff on the outer flanks at
Yeah.
the
Yeah,
Kamehameha Kanaloa, that there they just see thick mats of microbes where you can't actually see the fluid. You can't, there's no evidence of
Like
flow coming out,
subsurface.
but it's
yeah, for sure.
but there's like a chemistry delivery mechanism. So
Are
Very,
we good? Are
very
we good,
subtle
Data
flow.
Science?
yeah.
Yes, we're good.
Go wide video. Yeah, you do.
Yeah.
I'm just kidding. Hey, Megan.
Yeah.
I love the
Do you
colors
want
of
to
these
see it for the
maps.
last hour?
So
Sure.
luminous. Really good.
Yeah.
With
Yeah,
the
it's very
pink
bright.
as
With the-
well, it's like,
Yeah,
Yeah.
with the pink
I think
and
with the
yellow, it's
mushroom
really
color
pretty.
corals as well,
Okay.
it just really
I'm gonna
offsets it,
start
doesn't it?
moving.
I mean, just the staining of the rocks was already making for a nice backdrop, but then
Yeah,
to get
yeah.
the microbial mat in as well is
the mat, the coral and the
There we go.
Oh, no.
rock together. It's
Mhm.
great. Very aesthetic.
yeah.
If the navigation doesn't become over my head, I'll need your
Most
assessment on
importantly.
that.
Would you like a DVL reset?
Sure.
It looks like there's another really big mat to hit.
Yeah.
And an ice
Maybe
cream
we've
cone?
just come across that
And
region.
a
There's
Zoom
some
in
mat coming up
whatever.
ahead. Can we zoom on that as we go past it?
Yeah.
You can see it in upper, so it's kind of should be coming into our field of view
Also,
soon.
is this of interest? This weird little...
Yeah, let's.
You want to zoom
Uh,
in something here?
I think it looks like maybe
Let's
we've not quite
start
got
on
to
this
the
stuff
main
look
area yet.
first.
Oh, yeah, sure.
If you look at this pale bit stretching around
Go for
maybe.
Yeah,
zoom
it's in this
video?
crack
Go
here,
for
I think.
zoom.
That's.
Are we looking
And
at
then
this
we're just
pale
going to nudge
patch?
a little bit just above that.
Okay.
Can you see there's a little crack in the rocks and a hollow?
Yeah. I'm just going to do it really gently, because
little
otherwise we go flying. Oh,
I
you
see
got
shimmer.
stuff
Yeah,
there.
we
there's
have
shimmer.
shimmer.
All right,
Yeah.
let's head back down there, please.
Yeah,
Sorry.
alright. That's okay.
We ain't
That's
going
the
nowhere.
fluid flow I was hoping for.
Yeah.
That is amazing that you saw that.
Well, he is the vent whisperer.
Yeah, I
Look at that
know.
tiny
Wow.
little crack and upper that I saw.
Mary spotted it too. But
Alright, go wide video. We'll find a spot.
that is definitely a whispering vent, though.
Yeah. Well, confirm.
Or a whipsering.
Whipsering?
Whipsering.
Whipsy, the whipsy vent.
Whipsy, wispy, whipsy.
Well, there you go, Chris. That was your hypothesis that you were
Oh, we
thinking.
made it 10 feet.
Emission from the outside of the cone. Yeah.
Good job.
Solid.
Thanks for
Good job,
counting in
team.
feet, making
Solid
it sound better
transition.
instead of like this.
I didn't even
She's
put a
Canadian.
move in
She
yet.
can't help herself.
Mhm.
She's gonna switch units like 2 or
Yeah.
3 times an hour.
What? Oh,
Canadians.
oh yeah, yeah.
Mm.
No, I'm all mixed up when it comes to units.
So
They all
is
are.
that stuff must just be like coral that's had stuff grow over it, right?
I think it's
That
coral.
three-dimensional
I don't think
structure?
it's coral. I think
Isn't
it's
it fossilized? It's like been turned to lithified or something?
no. I don't
Something
think
else.
so.
All right, we'll look
I
at that
think
later
it's,
then.
Maybe venting things.
Teeny tiny chimneys.
Yeah.
Maybe.
I don't know. I don't think it's a coral though.
I was just wondering whether it was like dead coral that had stuff grown all over it.
No, we haven't seen any coral here.
Wonk.
Could we also
Other than
zoom
the
on this
anthomastus.
weird brown
I was gonna say,
squiggly thing?
And it looks
Um,
like
Might be a little
tubey.
far. Eh?
Are the mushroom corals not corals? Is that just
I mean, the mushroom corals are corals, but they don't have
Okay.
skeletons, so
No.
they're all soft.
I see.
Is this too far?
Depends on what we're doing. If we're
Yeah,
gonna
what
be
do we-
temp probing, we need to
Yeah.
get in a little closer.
Yeah, I imagine we're temp probing, right?
I think we're going to be
Yeah.
temp probing, and I should imagine Drinking Bird might be making a second appearance.
I would be interested in a
Yeah.
round 2 of Drinking Bird, but
Absolutely.
Let's temperature probe first.
I'll just sneak in here.
Round 2.
it looks pretty low temp.
Ah,
I
Chris
think it
Little
should
is
be good.
suggesting maybe it's a dead sponge.
I think it's, well, we haven't seen any sponges here either.
No, but Chris is the guru of fossilized stuff around vents.
Maybe
Going
they
back
will be.
hundreds of millions of years. So.
Is this area hundreds of millions of years old?
No, but
Is it hundreds
the
of
animals
years old?
the animals get turned to stone somehow to end up in the fossil records. So he might know what a thing that's been.
It doesn't look like a sponge to me either.
Doesn't look like any vent I know either. But
All right.
we'll
I
have
don't
it.
know. If it's not biology and if it's not geology, then it must
Heating up in
be.
here.
It's interdimensional space.
That's great.
It's just a portal.
Yeah, I'm just settling out.
great.
There we go.
Samantha's taking layers off.
I know. I'm already
It's getting
seeing
hot in here.
numbers.
We haven't even measured the temperature yet.
All right.
It's amazing what a difference
What
1.3 degrees Celsius can make.
Yeah.
do we want to
In
do
here
first?
especially.
She didn't even bring sleeves to
What
the
do we
party
want to
to
do
start
first?
with.
Science?
Yeah, I don't know
Temperature
how
probe,
she's
please.
mixed
Temperature
up
probe.
sleeves.
That's good.
The good old
We want to
temperature
poke it
probe.
with a stick.
Yeah,
All-
that sounds great.
all reliable, right?
Yeah, all reliable.
Can I give the temp probe a tattoo that says "all reliable" on the inside of it?
Someone really
Or
should
just-
scrape that
Or just
into it.
motivated. You just need some ink.
Motivated.
We have an engraver. That could be you.
Oh yeah. It'll stop the electronics working, but you know, but it'll look really
Yeah.
cool.
It'll make me
In fact,
feel
yeah.
better.
We'll engrave it, we'll stop it working, and then we'll challenge any of the other watches to find a hotter sign.
Yeah.
I'll love to see it engraved with all reliable.
That has to be all, not
All, absolutely, 100% all.
Oh, there's a squid.
Oh yeah. Thank you,
It's
Dim.
coming to visit.
Maybe it
right
likes the
down
hot
here.
bath.
Mm-hmm.
I could do with a hot bath after this
So-
dive.
Oh, yeah.
Mm.
It got cold again.
Okay, all reliable. We have need of you.
It's so cold in here.
Can we do a quick zoom again to see where we saw the shimmer, so we can?
Yeah, it's still there, I think.
Yeah.
Just above,
You can push in further.
to the right of where the logo is of the Nautilus, of OET.
Yeah. Can we go any
Okay.
further, or is that full?
I can go further.
Yeah, let's go further. We can always come back out once I've seen where to poke. Oh, I think it's actually.
What about here?
I think it's in the foreground. Yeah, I think it might be there.
Do you want me to focus
I
on
would
foreground
say
really quick to see?
This, but
Because
I don't know.
Yeah, I think we might be seeing through it. I was wondering at first if it was coming out of that crack, but
I
there's.
thought it was coming out of the crack.
yeah, I think it's still solidly coming out of that crack that we're hovering over, but I think
Yeah.
there is
I
some.
mean,
I
we've
wonder
got
if there's stuff
elevated
down here as
temperature
well.
right where we are. So,
Yeah, I would. Can we just go in down here
Yeah.
where the pale yellow is?
Yeah, so it's still climbing just with the temp probe, right?
Yep.
Can you go a
Oh,
little
go,
wider?
yeah.
Great.
We can go multiple. Oh, actually, yeah. Can we pan down with the camera again
Yeah. Just want to
and
see.
zoom again, please?
Okay.
So
Don't
it's
know-
leveled out at 7.65 in this location.
Yeah, I think in here, that little crack there.
Okay.
Let's try that.
Tilt up a little bit.
Are you okay with going into the sediment here?
Yeah, I think if we can get more vertical, so we won't just come back out of it again, because it's that goopy stuff. We already got to 7.5 just doing that.
That's sort of perpendicular to the
let's-
slope.
Let's hold there, thanks.
Yeah, definitely opened up the flow at the top now.
Well, it's going up again.
Yeah. And you can see that the flow is coming out, down towards the temp probe rather than up.
Well, it wouldn't be flowing down though, would it?
No,
Buoyant
but
flow
more
wouldn't flow
out.
downhill.
It's not
Mm-hmm.
it's not flowing. It's not flowing up strictly, I think.
Okay, well let's let this stabilize and we can keep poking around. But
Yeah.
we're up to 9 degrees, or approaching 9 degrees Celsius and climbing steeply. Yeah, we can try a few different positions with the temperature probe and then choose the best one for the drinking
I
bird.
think in this little space, drinking
Mhm.
bird is not going to be that spatially accurate.
Okay,
Anyway.
so just being over the crack generally will be...
Yeah, I think somewhere in this vicinity will be
Okay.
pretty good.
So we'll just do a little search just to see what the warmest temperature is that we can measure before we...
Yeah, that sounds good. That sounds good.
Oh, we've broken 10 Celsius. Still climbing.
Someone is asking, is this possibly sulfur?
I don't think so. I think this is much more likely to be iron oxide. But we'll bring some home to find out and make sure.
I'm a little I don't know if the rock right next to it is hard or not, but it just appears like maybe we're like pushed up really
Wedged,
hard against
yeah.
it.
Okay, I think we are stable there
Okay.
with temperature. So let's try a little higher up. Let's try
Okay.
in here, yeah?
Like here?
A
Or do you want
little
the
bit
top?
higher up, maybe?
Like
In this
here?
area? Yeah, try that.
That's as far in as that goes.
Okay.
It's definitely hotter. Or how far did the last one get to?
11.6.
11.6. There we go. Cleared it. I would love a cinnamon raisin bagel and cream cheese right now. It's like toasted, like the cream cheese, like super melty.
Oh, yeah. I could go for something with cheese.
Oh, you know what I was just thinking? Yeah, after Gabby said that, was on the German ship, they have a thing called Käsebrot, which is just freshly baked rolls, but they would bake a selection of different rolls every day, but the really good ones were the ones where they just sprinkled cheese on the outside and melted it like a little crust.
Oh, yeah.
Oh man, that
That
was
sounds
like
fantastic.
that was a really good reason to miss breakfast and just get up and just have fresh rolls like mid-morning instead with coffee and like 10:30 late breakfast.
Hard to beat German bread. It's pretty
Yeah.
great.
Second breakfast.
Mm-hmm.
Looks like 12.7.
Yeah. Okay. You want to do second
Yeah.
drinking bird here?
Let's get it in, and then we'll see where it is, and
Yep.
if we want to reposition. But it's a bit of a, it sort of goes where it goes.
Well, I think the
So-
nice thing is this crack is probably going to define how it comes up into the water where drinking bird's going to be.
Yeah.
So
Yep.
Okay.
there should be
Go
no bad
half wide.
answers.
Half wide.
Okay, yep, and I'm gonna stash it. You two are amazing. Like with the
What?
zooming and the panning all just happening in a coordinated effort is awesome.
I mean, otherwise I'm just
We are
sitting here
so close to
staring
the
at
other
it.
site. I don't think there's any justification for
mean,
filling
also
another
a
chamber
very
with microbes here.
important
I think the stuff
Yeah,
we had from the
no, I
first
think-
site is fine, but
Yeah, it looks
getting
the
the
same.
drinking bird data is
Yeah.
I love the little boots. That was such a...
yes. Well, couldn't have drinking bird on deck without his boots on.
steel
They're
toes.
steel-toed, yeah.
Yeah.
I also love how in bubble cam you can see the front view of drinking bird.
Yeah. I love bubble cam.
I mean, bubble cam used to be all we had.
I think it's my favorite.
It's a good camera. It's a shame that we keep it on such a small screen, you know?
It's so versatile.
Yeah.
Which one is bubble cam?
It's that
the-
bottom right
Oh, I
of that
see.
fore panel.
It's a good camera. Okay, drinking bird.
Come back out to play.
So I definitely don't think we'll be over the vent. Just FYI with where we're positioned.
I reckon if you put it out horizontal, and then we'll see where it is, and if we want to, is it possible? Yeah, move if necessary.
Okay.
Yeah, I think Gabby did a nice thing last time of get an idea of where it wants to be, and then let drinking bird up a little bit out of harm's way while we reposition, and then
Okay, I've got drinking bird. It's not in harm's way.
right. Did you want to bring it down a little bit so we can
Yeah.
see where it would be if we?
Yeah.
Because there's always the chance it would actually be just perfectly placed anyway if we're lucky.
This is getting pretty close.
If you look at where the front of the
Yeah.
porch is, then
Right on that
it's just touching that little bit of coral.
Yeah. So maybe we
So,
could hold
like-
it here for a minute just to get a stable reading there, and
Okay.
then see if we can reposition and get a slight bump, but I'm not sure we'll be able to hold it stably in the crack. But if you
think
can
if we
bring it
just
as
had it,
low
like,
as possible.
If it was
at
just
this
overhanging that edge of the rock, that would already be kind of in the right place, I think, for flow to be coming up around it.
How's that?
They got some nice signal there, look.
position. I think that looks good, yeah.